Hi students, here in this video I am going to explain you differences between the C3 and C4 plants which is given in the NCERT book. In addition to that one, I am going to explain some more differences also, and I am going to clearly explaining the entire differences why it came also in this video. Now you see differences why what is the C3 and C plants, that is they depend upon the environmental condition only they are adapted. Now you see he has given 16 differences in the textbook as in the form of the chart. Out of these things they are having internally so many neat creating possible bits are there that I am going to show you. Now first cell type which Kelvin cycle takes place. Kelvin cycle is compulsory for any type of the either it is C3 or C4, it is compulsory. So the Kelvin cycle can be taken place in the C3 plants in the mesophyll cells and C4 plants in the bundle sheath cells. That only C3 mesophyll and C4 bundle sheath cells. And cell type in which the initial carboxylation reaction occurs. Cell types initial, that is a carboxylation means carbon fixation, where it will be taken in the mesophyll cells, that is the of the C3 plants and C4 plants also. First fixation, carbon dioxide fixation, either it may be C3 or C4, it will be in the mesophyll cells of the plant. Now, how many cell types does the leaf has that fix CO2? For carbon dioxide fixation, how many cell types does the leaf has? The leaf has only one type of the cells in C3 plant, but two types of the cells is to present in the C4 plant, that is in the mesophyll cells and bundle sheath cells. The same thing, bundle sheath cells formation and this type of cringe anatomy that also given in the C4 plants. So only one type of the cells in the C3, but C4 is the two types of the cells, that is the mesophyll cells and the bundle sheath cells. Two places, and how many places it will be fixed, that also you can ask. That can be. It will be fixed only one place in the mesophyll cells of the C3, whereas in the C4 plant, it will be first fixation will be in the mesophyll cells and second time fixation will be in the bundle sheath cells before enter into the Calvin cycle. Remember it. This is also important bit, which is the primary carbon dioxide acceptor. Carbon dioxide acceptor first in the C3 plant RUBP, ribulose bisphosphate, whereas in the C4 plant phosphoenol pyruvate PEP, that is the phosphoenol pyruvate. Here he can ask one more thing. In RUBP used to take in the form of the gas state, carbon dioxide in the gas state, but PEP will be take the carbon dioxide from the bicarbonate that he has given clearly in the diagram, that is HCO3, that is the bicarbonate liquid condition. It will be absorbed in the primary, in the, that is the cytoplasm. All that entire thing I explained in my video, that is the C4 plants. If you see that one, you will get clear idea about the why it is in the liquid form, what is the advantage it is having, why it is doing like this, all the things I discussed. So here it is the primary acceptor RUBP and PEP. Number of carbons in primary carbon dioxide acceptor, and number of the means ribulose bisphosphate, it is to have the 5 carbon, it is a 5 carbon compound, whereas phosphoenol pyruvate is a 4 carbon, it is a Number of primary acceptor. 3. This is a 5 carbon. That is the first acceptor is PEP. That is a 3 carbon. And next, which is the primary carbon dioxide fixation product? In the product, whenever we are seeing C3 is in the C3, two molecules of the phosphoglyceric acid will be formed, and in the C4, oxaloacetic acid will be formed after taking the carbon dioxide. Then in the form of the carbon, that I allotted bicarbonate formation, it will take. Remember it, that is most important. And the number of carbons in the primary carbon dioxide fixation product. After carbon dioxide fixed, and in this one, that whatever the product is coming, how many it will have, that is the primary carbon dioxide fixation product acceptor. Okay, which of the primary carbon dioxide fixation product? C3 it is the phosphoglyceric acid, and C4 it will be the oxaloacetic acid, which is 4 carbon, and this is the 3 carbon. Remember it, this you are not. And the number of the carbons primary carbon dioxide fixation product, C3 will be the 3 carbons in the PGA, whereas C4 it will have the 4 carbons. Based on that one only they given the name. C3 means first stable compound which is formed in the Calvin cycle is, that is the C3. But first carbon dioxide fixation, first stable compound formed in the C4 plants is a 4 carbon compound, that is the oxaloacetic acid. Remember it. And the number of carbons in primary carbon dioxide fixation product C33 and C44. And does the plant have Robesco? Yes. The plant means either it may be the C3 or C4 plant, they will have the Robesco. Why? Because Robesco is the most important enzyme which is to fix the carbon dioxide and do the Kelvin cycle. In the RUBP will be taken one carbon dioxide and it will become the 6 carbon compound. Immediately it will divide into 3 carbon compound, which is the phosphoglyceric acid, and that is the, so P Robesco is important and it will be present. It is the, both will have the Robesco, but Robesco is present in the mesophyll cells of the C3 plants and bundle sheath cells of the C4 plants. That he has not given. Remember it. And which cells plant have the Robesco? That C3 plants will have the mesophyll cells and C4 plants will have the bundle sheath cells. And the carbon dioxide fixation rate under high light condition. High light condition will be followed by the high temperature. Because of that high temperature only, in the evolution C4 plants are formed, and so for that only is the, what it is, carbon dioxide, which of the carbon dioxide fixation rate under high light condition means high temperature also. C3 plants it will be low. Why? Because photophosphor, that is the photorespiration will be taken in this one. So it will be the low carbon dioxide fixation. Because it is undergoing, some of the molecules will undergo photorespiration. But in the bundle sheath cells, it is carbon dioxide fixation will be high because it is avoiding the temperature and oxygen percentage. That you should have perfect idea about the C4 plants. Then only you can understand the differences. Why it is not having the negligible, it is having the fast photorespiration, because the bundle sheath cells used to have a cover. There he has written clearly that it is impermeable for the gases. Means here you should know about the why they are, what the condition and the high temperatures. The stomata will be closed because of the ascorbic acid, abscisic acid. So the stomata are closed, inside the oxygen concentration will be increased. So the oxygen may enter into the bundle sheath cells. So it will be avoided by the thick cover which is present around the bundle sheath cells. So this is the oxygen cannot enter into the bundle sheath cells because of the thick layer, and so there is no question of the increasing of the oxygen, only carbon dioxide will be used. That is so it is which the cell plant have the PEP case. Now if you see, does the plants have a PEP case? No. The C3 plants, it is not having. PEP case means pyrophosphoenolpyruvate carboxylase. C3 plants won't have, and C4 plants will have the PEP because first carboxylation will be taken in the mesophyll cells with the help of the PEP, phosphoenolpyruvate. And next will be, which cells of the plant have Robusco? C3 plants will have the Robusco in the mesophyll cells, whereas C4 plants will have the bundle sheath cells that already I discussed. And carbon dioxide fixation rate under high light condition. This is the most important bit. This one. High light condition, what happens? In the high light followed by the temperature, that is the light and temperature are not different, both will come. So what happens? At the under high light condition, C3 plants will be doing the low carbon dioxide fixation, low, and in the sugar preparation also. Why? Because some of the cells RUBP, ribulose biphosphate, will be used for the photorespiration. That is the condition. So then the C3 plants, it will become low when the temperature is high. High light means high temperature also, remember it. And C4 plants, it will be high. What is this? Because C3 plants is where it is present. Whenever we are seeing in the crunch anatomy, the C3 plants used to fix the carbon dioxide in the Kelvin cycle in the bundle sheath cells which are present around the vascular bundle. And already some cells are present on the bundle sheath cells. So temperature on the whatever is present in the mesophyll cells comparatively that one, the temperature will be less in the Kelvin cycle, whatever it is, in the bundle sheath cells. And now, and one more thing is, so it is the high productivity, it will have the C4 plants, but C3 plants will be affected because of the oxygen becoming more oxygen will be taken by the Robesco enzyme, and it will be that photorespiration will be taken place. But C4 plants, it is oxygen fixation is taking place two places. One is in the mesophyll cells by the PEP phosphoenolpyruvate carboxylase enzyme, oxalic acid will form, and second one will be the Robesco in the bundle sheath cells. So in the bundle sheath cells, already temperature is reduced, one thing, and the next thing is oxygen is not there because the thick cover which is present around the bundle sheath cells. So C4 plants high it will be. Whether photorespiration is present at low light intensities, means low temperature, low light intensity. Temperature is more, then only C3 plants will reduce its productivity. But temperature is low, there is no. And one more thing you have to remember here, the thing is, the C3 plants are having the saturation point, that is the 460 microliters given in the factors affecting the photosynthesis. But C4 plants is having the 360 only, but 450 will be in the, that also will affect. Those things I discussed in the already in the factors affecting the photosynthesis. Please see that one also. You will get every question, whatever comes in the need from the photosynthesis, you can give the answer. So it will be high. whether photorespiration is present at low light intensities. Low light intensities, it is negligible because it won't be taken. Photorespiration cannot be taken. And next will be whether photorespiration is present. High light intensities, yes, high light intensity C3 plants will be high, means temperature also will be high. Photorespiration will be high. But in the C4 plants, already adaptation is taking bundle sheath cells. It is happening. It is negligible. Photorespiration almost will be avoided. So negligible. Whether photorespiration would be present, low carbon dioxide concentration. This is most important. One more. Low carbon dioxide concentration. When you are reading the C4 plants, the Revesco will absorb the oxygen if the oxygen concentration is more or carbon dioxide concentration is less. That he has given an NCERT clearly. So carbon dioxide is less. Naturally, compared to ratio, oxygen will be more. Then the Robesco will take the oxygen. So here it is whether proto-respiration present the high light intensities and whether the proto-respiration would be present the low carbon dioxide concentration. Low carbon dioxide concentration, photorespiration will be taken place as he has given in the C3 plants. The carbon dioxide ratio, carbon dioxide and the oxygen ratio, when it is changed by decreasing the carbon dioxide or increasing the carbon oxygen, he has given, remember it. And the next will be whether photorespiration would be present at high carbon dioxide concentration. This is also most important. Photorespiration would be present at high carbon dioxide concentration. High carbon dioxide concentration is there, then there won't be any change because oxygen percentage may be increased, but the carbon dioxide concentration also is there, the ratio won't be changed. That is the thing. And one more thing here, carbon dioxide increased. Which one will show the high productivity, either C3 or C4? What you will give answer? C4, C3 plants will give more because C3 plants are having saturation point at 460, 450 microliters. But C3, C4 plants are having the 360 only. That difference I clearly discussed in the other video, that is the factors affecting the photosynthesis. Please see that one. Because they are interlinked. The questions will come in the neat, not direct. Interlinked only will come. You have to prepare in that way. And next, so it is C3 and C4 plants, whenever we are increasing up to some extent level, it will be negligible. But if it is beyond the 0.04, what will happen if you ask? What you will give answer? C3 plants will be more. C4 plants won't have because 360 it is. 360 means that is nothing 0.036. That also I discussed in the factors affecting the photosynthesis. Please see that one. And next, temperature is optimum for C3 20 to 25, C4 30 to 40. This is also very, very important bit. Why? Think over it. Because C3 and C4 plants, both they will do the Kelvin cycle. So, Kelvin cycle won't be affected by the temperature, as usual temperature only. But the carbon dioxide carrying mechanism, C4 cycle, will be not affected by the temperature, and carbon dioxide freely it will bring. That also I discussed in the C4 plants. Please see. So C4 plants used to bring the carbon dioxide, that is the malic acid formation, oxaloacetic acid formation, malic acid formation. They won't be affected by the temperature. So they will bring very easily. But dark reaction, whatever the enzymes are there, they will be sensitive to the temperature. And one more thing I will see. So here they are not affecting the temperature, will be tolerating capacity will be more in the C4 cycle, enzymes, whereas C3 plants won't have that much temperature tolerance in the Kelvin, which are in the Kelvin cycle in C3 plant. Now the thing is, go to the biomolecules enzymes. Every optimum temperature up to that one, every 10 degrees centigrade, the reaction will become double. Later on, after optimum, but if you increases, it will fall in the same ratio. Given or not? It given. Now the optimum temperature of this C4 cycle is 30 to 40 degrees. So if it is 25, it will reduce. So for that one only, the optimum temperature, why it is 30 to 40, it is for the C4 cycle, not for the Kelvin cycle. Remember it. This he can ask the question. Now examples he asked. That is, here now you have to see a given. That is the only 8,100 plants used to belongs to the C4. Remember it. Means 3% of the gymnoangiosperms. Total 3 lakhs are there. It is 8,100 or 200 like that it is. What? C4 plants. And one more thing. So in this way, C4 plants, whatever are there, they are very less, but it is having some adaptation, which is positive adaptation for the environment. For example C3, neem, pepper, rice, wheat, oat, tomato. C4, that is the sugarcane, maize, sorghum, and amaranthus. This is the dicot. So it doesn't mean only monocots will show the C4. Some of the dicots also will show the C4 type of the cycle. Now, whatever I discussed, these things in the C4 cycle, I discussed completely. And factors affecting the photosynthesis also I explained detailedly. If you see along with this one, if you see those videos, you will get what is C4 cycle and the differences, what is the factors affecting. These are the most important for the NEET, which can be, he can create new type of the questions by mixing all the things. So, please see those videos also and prepare, note down all the points on one point and prepare for the 26 NEET. So if you like this video, please leave like mark and subscribe the channel. From the next video onwards, within 1 week I will start digital board. It will be more clarified, clearly you can see. So you next video or next video, within 1 week I will arrange it. You will get more clear board pictures under this thing in the from the within short time. So you subscribe the channel and see the previous, which is the having the more information. So subscribe the channel. Thank you.